So welcome back to another edition of the Heart to Heart podcast. And my guest today is Kevin McKernan. So Kevin is the CSO of Medicinal Genomics, and he also worked on the Human Genome Project from 1995 to 2000. So Kevin, welcome to the show.
Thanks for having me, Mike. Really appreciate jumping on this cast. I've enjoyed your work in the cannabis space. I've seen you on Joe Rogan and many other places, and it's refreshing to see.
Well, I'm really excited to chat with you today. I haven't had a guest, like you on this podcast before, especially with your background and what you did, and maybe before we kind of get into a little bit, you can give us just a little bit more of a background on what you've done in the medical cannabis space.
Certainly. So, as you mentioned before, I started off working on the Human Genome Project and getting familiar with DNA sequencing, and a lot of those tools were eventually being deployed on cancer patients. So we were sequencing the tumors of patients to see if they were druggable in unique mutations in their tumors that we could target that wouldn't necessarily have an impact on the person's native genome. So I built some DNA sequencers. The solid sequencer is one that's to market. Has been there for a while. It's fading out. I've also been involved in building the Ion Torrent sequencer. But these were mostly getting leveraged in cancer. And the one challenge in drugging cancer, as I'm sure you know, is many of the drugs we have to choose from are pretty toxic. And if you can personalize the treatment, it doesn't help if the drug profiles are fairly toxic in that regard. And that's when people turn my attention to cannabinoids. The toxicity profile, as you know, is quite tolerable, and there is some promise with these compounds in reducing the size of tumors. And that was really intriguing to me. And so at the time, I had a lot of non-competes with the companies I was involved with, and decided that one field that they probably couldn't claim any competition in was in the cannabis space, given this is around 2010, given the nature of its legality at the time. So I decided to jump ship from those organizations and begin sequencing cannabis genomes to better understand what genes were involved in synthesizing the various cannabinoids in the plant. There's more than one, and it's not all about THC when it comes to cancer. So we started putting those genomes public to try and build like a Linux operating system, if you will, of pharmaceuticals that aren't necessarily entrenched in a lot of this FDA and regulatory thickets that we see going on today in even the COVID landscape.
Interesting. So basically, Kevin, what I kind of gathered there is that, you're involved in the Human Genome Project, looking at, drugs that would help cancer patients. And, as you mentioned, and as we know, unfortunately, the drugs for cancer are not always the most effective. Sometimes, they're very effective, but, sometimes they're not. So, you kind of did your work there to make sure that the selected patient was getting the proper treatment according to their DNA, it sounds like. And then you sort of took that idea and now have applied that to cannabis to ensure that people who are using cannabis are using it in a way that that fits their DNA, and that makes sure that they're taking the right type of cannabis. Is that accurate?
Yes, and we're not in the business of drugging patients at the moment. Most of our focus right now is building genomic picks and shovels, if you will, for the cannabis space, so they can breed for specific chemotypes. If you want a plant that breeds specifically, let's say cannabidiol, which has some publications in prostate cancer, that's actually hard to find right now in the dispensary market. It's a very rare cannabinoid, and you need to know how to breed for it. There's a few organizations that recently have bred for that, and you're starting to see a compound called cannabidiol or CBG filter into the marketplace. Prior to this, you probably saw a big focus on CBD because it was showing some impact in epilepsy, but it's also been well published by many people in the space, from Christina Sanchez and many others, that CBD also plays a role in reducing the growth of some tumors. So we don't yet know which of all of these cannabinoids need to be deployed against which particular tumors. That's work that's kind of ongoing, and I think you'll probably see Didier, I'm going to murder his last name, but there's a gentleman who presents at CanMed quite a bit on applying these different cannabinoids against cell culture to see which types of tumor cell lines are most impacted by different cannabinoid profiles. And we kind of think that's the future of where it's going. But it's right now, we're at the stages of the market is so early that we're just in the game of sequencing as many of these genomes as possible and discovering the markers that predict which plant is going to produce which cannabinoids, so that they can effectively breed for plants that specifically make one versus another.
Okay, and I'm really happy you brought up CBG because recently I've done some other videos and things on CBN, and I've hesitated to chat about other cannabinoids in the past, just other than THC and CBD, just because they're not commercially available. So, people
Right.
sort of get frustrated. And, I was talking about, THCV, for example, like back in 2016,
Yes,
2017.
that's another good
And I
one.
mean, it's still not commercially available. So, I don't talk about it with people. But CBG, I think, is one that is becoming more commercially available. It's not certainly not, as popular as THC or CBD, but it seems like and for people listening, I mean, there's over 140 cannabinoids in cannabis, as far as I know. You may have the exact number. And we know that there's some effect that THC and CBD has on certain cancer tumors, and there's been some promise in some preliminary research, animal models, and some case studies. We don't have a ton of like robust randomized controlled trials on it
Right.
yet, but it does seem that there are various different cannabinoids that do have anti-carcinogenic properties. So it kind of sounds like also too that you mentioned CBG and prostate cancer. So it sounds like you have to get the right cannabinoid for the right cancer. So how did you guys find out that CBG is potentially the right cannabinoid, say for prostate cancer, over THC and CBD, or maybe in addition to? Like, how did you guys determine that?
So that wasn't our work. I'm just I'll have to forward you some papers on that. We can link to
Okay.
the notes of the show just to clear that out. And it was David Mieri or Dede Mieri who did some of the cell culture work. So I think you've hit on a really important point, which is most of the clinical trials on this are limited right now due to production. No one wants to engage in a clinical trial with CBG if there's some background THC or CBD that are also present. You'll also notice there are fewer trials going on with the acid forms of cannabinoids. This is what the plant actually makes all of these cannabinoids with a carboxyl group on them. However, it's unstable and it decays. So CBDA tends to decay into CBD, and THCA decays into THC. The same thing's true with cannabigerol. The plant makes it as CBGA and then it decays into CBG. And so unless you have these things really purified in one form, and they tend to be the decarboxylated forms because no one likes to put medicines through trials that are not stable, that could change over the course of the trial. Unfortunately, there's a large section of the cannabinoid sciences that aren't making it into clinical trials because of stability artifacts with the compounds. And we seem to see a selection of trials going on with the ones that are decarboxylated, because that's kind of an endpoint. You can heat them all to get them 100% decarboxylated, and then you have a stable molecule. So you'll tend to see the trials are first limited by the fact that you need these hyper purified preps, because the FDA really doesn't like these botanical drug substances. They have a pathway for that, which DW took, but it's more complicated than putting a single purified molecule through a trial. So you tend to see some reductionism going on, which isn't always what is favored by the cannabinoid physicians. They're fully aware that that reductionism sometimes leads you to Marinol, which wasn't very effective. Once they took THC out of all the other constituents, they found less effective dosing than they found with more full spectrum plant medicine. So
Just to interrupt there quickly, Kevin. So Marinol, is that very similar to Nabalone?
I believe it is. I think
Okay.
it's just the different marketing name than the Nabalone, if I have it correct. But
And just for the people listening, before you go on there, Kevin, Nabalone is a prescription drug in Canada. It's been available for at least 5 or 6 years, I believe. I know there's one trial that showed that it may be effective for reducing some nightmares in PTSD patients that came out a while ago. But for the most part, and I say this speaking as a physician who's prescribed cannabis to a lot of patients with PTSD, most people will tell me that they do much better using dried cannabis or some type of form of full spectrum cannabis that has all the cannabinoids and terpenes, as opposed to just using the synthetic version of Nabalone, which is just THC only. So just wanted to point that out for the people listening.
Yeah, and you do see that emulated in what GW Pharma has done with Sativex. I believe that one's actually a 50-50 mixture of CBD and
It was.
THC.
Yeah.
And when you get into even Epidiolex, it's not 100% CBD. It's like north of 90%, and there's some other trace compounds in there. So this is one of the, I'd say, paradoxes that we live in the cannabinoid sciences, is that the FDA tends to, I don't even want to blame it on the FDA. I just think the process in general tends to reduce these into single compounds because it's very difficult to study these multivariant drug formulations. But that does tend to weaken the impact that people see when these things get FDA approved compared to their natural counterparts that we might see. The flip
And
side of that
also
is,
too, I imagine that because there's, you're not able to do the clinical trials that you want to do, then unfortunately that's going to, set back the timetable for these medicines becoming available.
that is, and I think there's a second impact that's happening with a lot of the placebo-controlled trials, is that I can forward you some literature on this, but there are links to the placebo effect being driven by the endocannabinoid system. Right, so this makes it somewhat complicated. If the pathways that you're targeting are known to be the pathways that are elicited through placebo effects, your placebo-controlled trials can sometimes appear a bit slanted. And I think that has happened in some of the pain studies where cannabinoids rarely show themselves to be beneficial for pain in some of the studies that were put through with single cannabinoids. But those are done next to controls, and the placebo controls are often elevated. And they've shown that that's linked to anandamide synthesis, and that the placebo side of this may need to be reconsidered. But at the moment, we do have to live with these randomized controlled trials. Otherwise, it's all observational, which I think observational are very helpful too. If you have enough, the numbers are large enough and you can coordinate them, observational trials can be extraordinarily powerful, especially when you're dealing with compounds that we know have really good safety profiles and have thousands of years worth of human dosing. But the gold standard still seems to gravitate toward these randomized, controlled, double-blinded studies.
Yeah, I agree with you though that the randomized control trials are great, but it definitely makes the research more robust and better overall when you do have epidemiological evidence or observational evidence and anecdotal evidence even as well, to case studies, everything. I think that it's good that we're putting a lot of emphasis on randomized control trials and considering them to be the gold standard. But the problem is though, and we're kind of I've already kind of discussed a little bit, is that it's the waiting game, and it's that if you have to wait until all these randomized control trials come out, how many people could you have treated in the meantime,
Right, right.
safely and effectively? And that's something that I think we need to take into consideration, a little bit better, especially too when you're talking about something like cancer, with where we have
Shot clock.
very limited options for certain types of cancers. Certain types of cancers are very, very treatable, but there's other types of cancers that are not very treatable at all. We don't have good medications and good treatments for them. And for those types of patients, I think that using something that is, relatively experimental is something that, a lot of patients would welcome. I know that for myself, if it was, you can do nothing or you can take something that may work, well, I'm going to take my chances on the something that may work as opposed to absolutely doing nothing. And so I think that we do need to try to increase the efficiency of getting these trials out there. And also we need to consider the evidence that we have outside of randomized control trials and take that into consideration when we're making clinical decisions for patients who are literally dying.
No, you've hit on, like, I think the critical point that I think is a precedent the FDA is against right now, which is when there's a shot clock like that in cancer, the rules should change. And I also think when the toxicity, I understand you want these placebo-controlled trials to protect against something getting out that may have a toxic effect, so that you violate first, do no harm, right? But when you get involved with cannabinoids and the toxicity is so safe, that there should be a different path through the FDA for those types of medicines, because there's nothing wrong with the placebo effect if it in fact helps the patient. I mean, who cares how it happened? psychology is a powerful thing.
Agreed.
And so if the medicines, in fact, this comes down to a lot of the right to try legislation. I've experienced it firsthand. I had my father had a prostate cancer get out of control before they diagnosed it. It was stage 4. And you know, they never give you length to live, but it didn't look like he had a lot of time to live based on it having he had bone mets everywhere by the time they discovered it. We managed to keep him alive for 3 to 4 years on cannabinoids and a lot of it was just, we believe, was palliative. And if we got any of the antineoplastic properties of cannabinoids, that was just frosting on top, because he really couldn't eat. And he couldn't keep any of the medicines they were giving him down. So we had no idea whether he was actually getting the right anti-hormone treatments that the hospitals were giving him. They started him on Casodex, which is an oral drug with a long half-life. And the moment your patient throws up Casodex, you've got no idea to know how much more to give him. How long was in his stomach, and it's got a long half-life. Do you give him another dose? Do you give him half a dose after they've thrown it up? And the cannabinoids helped him keep all of the other meds down so he could get the appropriate therapy the hospital was asking for. And the hospital was, a lot of the hospitals we work with in Dana-Farber, they were very forgiving on the fact that we were using cannabinoids. They were very understanding. They've seen this work, but that's obviously not in their guidelines to necessarily prescribe it. And that was a very reassuring thing to see, because there were many hospitals he went to that nearly threw him out for using them. They just wanted nothing to do with patients that were deviating off their plan. So I have to commend Dana-Farber for actually being open to it. It's not always the case that you get that, but I agree with you that I think these medicines need to be looked at in a very different light, considering the urgency that the patient needs them and the lack of toxicity that's present. The placebo effect is really, to me, irrelevant at that point, because even if my father was benefiting purely out of a placebo effect, I don't care.
Yeah, I don't think the patients or the family would either. I mean, as long as they're getting better, I mean, that's ultimately the goal of treatment. So whether it's, you know, through cannabinoids or through placebo, then you know, as you said, like, who cares? And to your point too, I think it's really important to note, like, I don't know if I ever really thought of that before, you know, thinking that, you know, patients using THC are able to keep, you know, their other medications down because they have THC on board. So it's like, you know, THC allows them to use their medications,
Right.
which is pretty incredible.
They went through, he had to wean off of his cannabinoids. We had him close to 300 milligrams, 200 milligrams of CBD and 100 of THC at this time, which is a lot of cannabinoids. Like,
Yeah,
and he was not excessively psychoactive, if you will. However, when his PSA levels started to go down after about 6 months of his treatment, he got very sensitized to THC. He started getting really stoned, even with 5 or 10 milligrams. It was uncomfortable for him. And so we started to wean off of that. But at one point, when he was very high, there's a whole other theory we have behind that we discussed with Christina Sanchez at one point. But we believe when you have lots of prostate-based tumors in your body, they're known to hyperexpress cannabinoid receptors, and we think they become a sponge for a lot of the cannabinoids. And when they start to shrink, you no longer have that sponge, and they end up going to your brain, and you can feel them. But that's just a theory we've read throughout the literature. But
interesting theory, though. I'm glad you mentioned it.
Yeah, yes. It tracked with his PSA levels. As his PSA levels went from 170 back down to like 20, he started getting really stoned on those high dosages. And so we weaned him off. We left the CBD in there, but weaned him off the THC. But they had to take him off of these things, or we decided to take them off of this when he had to go get a pacemaker put in. His pacemaker wasn't MRI compatible, so they couldn't scan him for a lot of the brain tumors that were starting to occur in his skull. And so weaning him off of that was, took about a week to dial that down. And then once he got into the procedures, he started vomiting and throwing up. And there's nothing they had in Manchester Hospital that could stop the vomiting. And so the nurses finally said, if you think you have this nailed with whatever you're doing, step outside and we'll turn the other eye, look the other way. And we had him back in the vomiting under control in 45 minutes, and they're walking him out of the hospital after that.
That's incredible.
So it was there's nothing. Interestingly, they didn't have Marinol in the pharmacy. That kind of shocked us. They're like, yeah, we just don't use it enough to keep it around. So because we said, look, if you're not comfortable with us using these oils, then let's take something you have in your pharmacy that should do the same thing. And they didn't have THC present there. So that, we ended up getting him oils, and those ended up getting the vomiting under control, and he was right back in step, and then had a good pacemaker. So that's something that's really shocking when you see patients that have that much of a physical effect. You can see them retching and vomiting and uncomfortable, and then in 45 minutes it just goes away. After
Yeah,
this
the
happens.
antiemetic effects of THC are incredible for a lot of people, and like you said too. I mean, depending on how you take the medicine, but particularly if you inhale it through a vaporizer or smoke it, and hopefully you're
Faster that way. Yeah,
vaporizing instead of smoking. The onset is so fast. Even with the vaporizer, it's going to be 5 minutes or less. So
right. We
you're
couldn't do
literally
that
going to
then.
get
Yeah,
symptom management is going to begin within 5 minutes of using the medicine or less.
yeah.
So it's I imagine then after 45 minutes when it's fully in a system, then his symptoms are significantly better managed.
And you're bringing up an important point about routes of administration. We could not use vaporizers in the hospital. We could give them oils in like applesauce, and that worked out for him. And it would have been a faster relief if he had, if he went the inhalation route. But we had other complications, I'd say, on the inhalation route, and that he was not a smoker, so it was not easy for him to like learn, inhaling vaporizers. And he was immunocompromised, so he was getting thrush quite a bit. And we were not confident in the sourcing of cannabis to know whether there was microbial burden present for vaporizing. I think combustion is a little safer in that front, which you rarely hear people want to talk about, because combustion is just thought to be like tobacco smoking. But there are risks of molds when you start vaporizing. The risk of mold exposure goes up, and so that's an area that I actually spent some time in. The backdrop is
I'd love to hear your. I don't know if you plan on chatting about that today, but I'd love to hear your thoughts on vaporizing versus smoking, and I'd love to hear if you have more thoughts too within the vaporizing. Are the cartridges as safe as dried cannabis in like a PAX or whatever vaporizer you're using? So do you mind touching on that,
not at
Kevin?
all. So that is a complicated topic, and the whole Valley crisis, I think, got everyone to rethink it a bit, which is helpful. But so generally, when you're extracting cannabinoids, you're decontaminating them from microbial burden. However, you might be enriching them for other chemical burdens, like myclobutanol and pesticides, and maybe even this vitamin E acetate that showed up in a lot of the vaporizer pens that they attributed some of the Valley crisis to that.
It seemed sorry, I don't mean to interrupt, but from my understanding, it seemed like they attributed basically 100% of the vaping scare to vitamin E acetate. Is that
I
correct?
think, yeah, and I think that may be a little premature. It does seem like it was correlated. In retrospect, I feel like it was so close to the COVID outbreak that I'm starting to question whether, how much of that overlap with respiratory infections. For instance, there was, they both have, ground glass opacities as one of the concerns, and the timing on the COVID thing seems to keep getting pushed back in time. That it may have been around in August of 2019. So I don't know if it's 100% vitamin E acetate right now. That might be a little bit mixed with some respiratory infections as well. But they didn't think that they were contagious. They did look for that, but with asymptomatic patients, it's hard to really measure contagion like that, right? You could have other family members who didn't. They made it look like it wasn't contagious when it was. When it was, from a PCR standpoint, maybe they could have seen it. So I don't really think there's a firm handle on the Valley crisis yet, in light of everything we saw that came after it. But certainly I don't suggest people run around and put vitamin E acetate in these vape pens when it's not needed. You can put other oils in there, I suppose, or not cut them at all, and not run into those risks. But I think the important thing to consider, and Ethan Russo's published on this quite a bit, that when you do extract these cannabinoids, you enrich. For the cannabinoids might go from 15 to 20% in the flower up to maybe 60 to 70%, but some of the other smaller molecules get enriched much more aggressively. Like myclobutanol can go up 100 times, and I think Ethan Russo showed a couple of these other pesticides that can get enriched 1,000 times in the extraction process. And so you got to make sure there's testing involved after extraction, so that those other compounds aren't a concern. But assuming all that is clean and properly quality checked, that is probably safer route of administration than vaporizing flower, because we have seen in the clinical literature aspergillosis happen to immunocompromised patients. I don't think this is a big risk to people who aren't immunocompromised. You may not always know when you are or not immunocompromised, so that's just something to consider. But Remington et al., I think, is probably the best paper that shows people. They went and did PCR analysis and sequencing on the aspergillus in the patient's lungs, and they found the same strain in their vaporizer. And so the thinking is the temperature upon which those things vaporize isn't always sterilizing the spores. And if you're immunocompromised, you can increase your aspergillus load. You're getting aspergillus daily when you walk around. It's fairly ubiquitous. So most people slough this off without a problem. But if you have diabetes or other immunocompromised conditions, you can get this in the lung, and that has a 50% mortality rate once it's in your lungs. If you get aspergillosis as an infection. So it's a very rare risk. I don't see a lot of, there's maybe 10 cases of this in the literature, and it's hard to know how aggressively people are looking for this because not everyone goes to the hospital waving a flag saying they just smoke cannabinoids. So I don't know how well it's being monitored, but there is evidence of this happening in the literature, and that's really the only risk I've seen in cannabis. I've not seen any fatalities that aren't microbial related.
I believe you can get it, again, probably even rarer than from cannabis, but from peanut butter as well.
Well, there you go. Yeah. So you got to put these in perspective.
But just, quite simply, just to kind of put you on the spot, like, so if you're a medical cannabis user or even just, a recreational user, do you think that you should be doing vape cartridges? Do you think you should be doing dried cannabis in a vape, or do you think that you should be smoking, or do you not even know what the safest route is?
I think as long as things are quality checked, you can use any of them. I think that's mainly the take-home message, is make sure it's tested, particularly if you're immunocompromised. So we wouldn't give any of those things to our father because we knew too much about the industry. In full disclosure, I'm in the cannabis. We sell PCR testing tools to the cannabis space to look for these things. So, take that for what it's worth. But we knew enough about how the Massachusetts market, and he was in New Hampshire and Florida, and some of those markets were less mature, that we're like, look, we just don't know what the microbial loads are on some of the plants that are coming out here, particularly the CBD lines. That goes under the hemp legislation, and that's less aggressively tested. The medical material is usually fairly aggressively tested in each state, depending on your state. I think Oregon's a little lax in their regulations, but other than that, most other states have come up to speed on microbial testing, and so they perform some level of Aspergillus screening and some level of total yeast and mold testing, so that the flowers that are coming through aren't contaminated. In which case, a flower vape is fine, and extracting it's probably fine because they're testing for pesticides in that process as well. It's when you start getting involved in some of these unregulated cartridges that float through the marketplace where all bets are off. You really don't know if there's vitamin E acetate in them. You don't know if they've been pesticide screened, and you don't know if the flowers are necessarily clean.
Yeah, it seems like to me, like, the first thing for sure is to make sure that you're getting a high quality product that's been, third party tested. And then, from there, it does seem like, if you're in a situation where your product can't get tested for whatever reason, and, hopefully you're never in that type of situation, it seems like maybe the biggest danger is related to the vape cartridges, just because, I guess that seemed to cause the most damage that we've seen when they've been contaminated. But that's certainly not to say that you can't get something very similar from smoking something that's contaminated, or vaporizing something, dried cannabis that's contaminated.
Yeah, there was a case in Bakersville, California, where I think it was in a bunch of pre-rolls. One patient got a cryptococcal infection. And they were continually going to purchase pre-rolls at a particular dispensary there. And as many people know in the industry, the pre-rolls are kind of what people sweep off the floor. They're the remnants that get shredded up and thrown into a lot of the pre-rolls. So it's usually not the highest quality flour that's going into those. And that person happened to get negatively affected. It wasn't fatal. They were able to address it, but it's another microbe that's not specifically screened for. We hope to catch that in some of the more broad scale. So some of the testing in the cannabis field is different in each state. Some states have total yeast and mold testing and total aerobic count testing. Those capture broad categories of fungi and bacteria, but they have enumeration limits that are set pretty high. there's 100,000 bacteria that are allowable in a total aerobic count, and usually 10,000 that are allowed in a total yeast and mold. But you don't know what they are. And oftentimes they can be benign and even beneficial microbes that are present, but they still flag the test and fail product, unfortunately. We're working in a lot of states to try and narrow those to be very species specific, so they target what are actually known pathogens, because there's a bit of a weird feedback loop that goes on when you aggressively test for broad scale microbes without any speciation. You take away a lot of the tools the farmers have to reduce the need for pesticides. And so if you take those tools away, you find more pesticides showing through, and not all the pesticides are on the list. They have a list of maybe 60 to 90 pesticides in each state. So they venture off and use other pesticides that are less known. So we're not really big fans of these broad scale total aerobic and total yeast and mold tests. They exist in some states, but there's no clinical utility to them. They're a bit of a tarot card. And if you have really high loads, they just don't like the product and they throw it out. But since they're not speciating the bacteria or the fungi, you don't really know that it's harmful. And when you put in tight regulations like that, you often induce other secondary effects and bad behavior. So we've been a proponent of really looking at clinical literature of things that have hurt people and test for only those things. Otherwise, this boy in a bubble world is kind of unrealistic. These are plants that grow outside, and a lot of these microbes are actually endophytic. They live inside the plant. You can't really get them out. So it's hard. It's impossible to be growing plants without microbes.
And just coming back, I know, we have about 20 minutes or so left here, and we want to chat about some things other than cannabis. But it seems like, Kevin, like you know quite a bit about which cannabinoids that we believe anyway, or maybe there's certain cannabinoids that we know can affect certain cancers specifically. And you mentioned there's preliminary evidence that CBG can be helpful for prostate cancer. Or is there any other cannabinoids, like can you speak of THC or CBD or any other cannabinoids that seem to be effective for any particular cancers?
So I have point people to a lot of the videos we have on the CanMed website, because those physicians get up and they share anecdotes, and particularly the glioblastoma cases seem to be benefiting from a full spectrum. They've been, and not even necessarily high doses. I think Deb Kimlis has some presentations on there where she's had, granted, these are all case studies of a couple patients. So these are not large randomized controlled trials, but these are just what physicians are seeing with their patients. So I'd point you to Deb Kimmel's work. I'd point you to Bonnie Goldstein's work. She's presented on some case studies there. There's also been some great work on autism and ASD and the more pediatric kind of epilepsy and neuroplasticity patients. That population's been looked at quite a bit. They usually have some case studies on the website there. Christina Sanchez gave some interesting work on breast cancer as well. And I think in the breast cancer field, I think is important to look through a little bit more cautiously, because there's different types of breast cancer, and we shouldn't be, I think you really need to type those breast cancers to know how hormone sensitive they are, because you may take a different approach based on this. So I think this idea of cancers based on the tissue of origin is starting to melt away, and we're starting to look at them as like, what's the molecular profile? Are they HER2 positive? Are they, what mutation do they have? Because that's more of a signature for the tumor than necessarily the origin, the tissue origin. And that, I think, that played a role in our father's case. Our father had a prostate cancer that was unlike other prostate cancers. Most other prostate cancers are an androgen receptor mutation. He didn't have those. He had something that was known as an AKT1, it's something in the AKT1 pathway. It was actually a BRAF K601E variant, which is a long, complicated word for something that's in the AKT pathway. And we sequenced his tumors and with Foundation Medicine, and they pointed to AKT inhibitors. And it happened to be that cannabinoids are potent AKT inhibitors. So we gave him both THC and CBD in that case, because they both had literature supporting suppression of AKT1. So I think if you can get your tumor sequenced, I think that's the place to start. So you know what type of tumor you're really dealing with, because oftentimes the tissue of origin isn't enough of a signature. And then you can then go from there and search the literature for which cannabinoids have been shown to inhibit those pathways.
And I'm glad too that you brought up breast cancer, just because I know, and HER2 positive, because I know that I've seen a study indicating, and I want to, I need to be careful about this. So, for people listening, check this. But I believe that there was, a study showing that a combination of THC and CBD, or maybe it was just THC alone, could be effective for that particular type of breast cancer. But there's other breast cancers where it showed that
It
it
can make
could
it
have
worse.
actually make it worse. Yes.
Yeah. I
So
think that's, we do have
it's
to be
really
careful
important.
there. Yeah, I think that's very key to look at that. And the other area that I think is worth some active discussion in the field is how did these compounds behave when you're taking an immunotherapy approach? That's something that approached us with a lot of caution because they were pushing down an immunotherapy route, and it's not clear that the cannabinoids help in that circumstance because the immunotherapy is really meant to rev the immune system up to fight the cancer, and the cannabinoids tend to modulate that back down to homeostasis. And so I think I've seen a paper too suggesting the cannabinoids might actually interfere with an immunotherapy approach. And so you've got to be a bit.
Yeah, and I've seen the same thing actually, at least one paper indicating that vitamin C sometimes can do the same thing as well. It can interfere
Oh,
with some
really?
of the medications that are given to people in cancer, because there is some legit evidence that extremely high doses of vitamin C can be effective for certain types of cancers. But again, it's probably the same thing with cannabis, is that it may have some type of negative, immunomodulatory effect, whereby it makes the other medications ineffective. So we do have to be careful. So like you said, I think the most important thing, and why your work is so important, is that we need to do the sequencing and get the genetics for these types of cancers, know exactly what we're dealing with, and then it's only then when we get the very, very specific type of cancer that we know that we're treating, like you did with your father, then you can go ahead and say, okay, I know that we have this type of cancer. I know that these cannabinoids have this mechanism of action. Therefore, it's going to be helpful. Or in another situation, you may be in a situation where, we have this type of cancer. These cannabinoids have a mechanism of action that is not going to be efficacious or helpful for treating this cancer, and may in fact be detrimental to the entire treatment plan.
Well, I think that the take-home message here is try to have physicians involved in this discussion because there is a tremendous amount of information on the internet about how wonderful cannabinoids can be on cancer. However, it is very complicated, and there are a lot of things that we didn't know about when we were looking at this with the father. And we were thankful to have a lot of physicians who did understand cannabinoid medicine. Bonnie Goldstein was very helpful. Dustin Sulak was very helpful. We had a good outreach to the cannabis community, and we knew all the cannabis physicians. And one thing that they would bring up, which I think is quite important to have on the table, is you really need to look at all the other drugs that they're on when you do this, because
Yeah.
the cannabinoids are known to interfere with drug metabolism, particularly CYP3A4 and CYP2C9 and 2C19. These are enzymes that metabolize a lot of other, like heparin, for example, is metabolized at these pathways. So it can change, warfarin and blood thinner medications. A couple of the chemotherapeutic agents that they were using were also metabolized through these things. So we had to be aware of the fact that if we're going to load the patient up on cannabinoids, we might need to, that might mean the plasma levels of the other drugs are going to persist for longer, and they're going
and
to overdose them on their other drugs.
and not necessarily just cancer drugs. Like it can affect anti-epileptic drug levels as well. So
Yes, indeed.
it's not certain, it's not limited to cancer drugs. So you do have to get a good medication history before you proceed with cannabinoids. And Kevin, I hate to cut you off there in a sort of switch topics, but and I mean, you're probably going to have to come back on and to chat
Oh,
about
I'd love
cannabis
to.
again, because there's so many more things that I want to chat with you about cannabis with. But I know that you've been pretty vocal on your Twitter about COVID, and obviously you have a very strong science background. You've mentioned PCR testing, a few times. So with that being said, with COVID going the way it is right now, like what are the main issues that you feel need to be addressed in this COVID crisis that we're going through?
Well, I'm glad you brought that up. So PCR is something close to my heart. We've been using it in genomics my whole career for 25 years. And the real tricky thing with PCR is it's great at detecting single molecules of either RNA or DNA, but unless you know precisely how to use it, you really can't discern whether that molecule is infectious or not. And I think that's plaguing the whole coronavirus story right now, is the news is incessantly following cases, and the cases are being measured by PCR somewhat sloppily. The PCR platforms that went into place went in so quickly that they never really calibrated them for what's known as viability PCR, where you actually measure whether the RNA is coming from inside of a capsid of a virus or whether it isn't just free floating in your system. So the biggest problem with PCR and COVID testing right now is that you're usually infectious for about a week, but you can be RNA PCR positive for 5 weeks. So 5 times the number of people get trapped in the PCR testing that aren't infectious. And so that has a huge impact on society in terms of categorizing the wrong people as being required to be quarantined, or pulling nurses out of hospitals because they're positive when they're not infectious. But it's also having impact now on us understanding whether these vaccines are working or not. Because now people are getting the vaccine and they're getting positive afterwards, but they're probably less infectious, would be my guess. The vaccine, if it does what it claims to do, it's a non-sterilizing vaccine, right? It's not meant to prevent the virus from getting into you. It's there to prevent you from going into overdrive and having a cytokine storm and have your system respond to the virus entry the second time a little bit more effectively. So we have this Delta variant scariant wave going on right now, which it's certainly there in a lot of jurisdictions and rising, but I think we're going to see a separation between hospitalization and cases, unlike the other waves. We should, at least, Iceland is having a huge spike right now. They're 95% vaccinated, but it's a PCR spike. The hospitalizations haven't shown up yet in Iceland. They are showing up in Israel, and that might be because Israel had less pre-existing immunity. Israel didn't get hit very hard early on because they had such an aggressive lockdown. And so that may be coming back to haunt them. We'll see what happens in Australia and New Zealand. They had really aggressive lockdowns, but they're in the Pacific, and they may have had some pre-exposure to this because everyone thinks it was circulating before December. So I don't know. We'll have to see if the case rise in those really tightly locked down countries translates into increased hospitalizations and deaths. But it's going to be different, I think, in all the jurisdictions. And unfortunately, a lot of this is just due to PCR not being used properly. My biggest
and Kevin, just to interrupt there for a sec. So, like, roughly speaking, then only maybe about 1 out of 5 people who are testing positive for PCR are actually infectious.
That's probably a good estimate. And I can point you to a couple JAMA articles that go through those numbers where they show you about a 7-day window of infectiousness and 35 days of which they can detect this. I mean, there's a couple methods that are out there that aren't getting deployed, I think, due to financial reasons in the testing space. But you could test people twice, which is a pain in the ass. But 2 days apart or a day apart, if the viral load's going up, you're probably on the infectious side. And if the viral load's going down, you're probably on the clearance side. And you could segregate the population that way as to whether they're infectious or not. You can use rapid antigen testing as well, which tends to capture mostly the people that are infectious. Or you could use these viability PCR techniques. I think the reason it's not getting deployed is, one, logistically, it's hard to do it twice. The other reason I think it's not getting done is that when you capture 5 times the number of patients for PCR testing, because you're very sensitive and picking up the people that are clearing it, all of their relatives get contacted for contact tracing, and that brings in a whole other stream of revenue for the testing companies. So for them to shrink this, even though it sounds like they'd be double testing and they get more money, they're actually getting less because they capture 5 times as many people in the positive category, and they get in the contact tracing information and testing when they capture a wider swath of people. So the testing industry is not motivated to sharpen the pencil on this. They want to get as many positives as possible so they can pull in more through contact tracing. So I'm not optimistic that they'll switch to a viability PCR approach, which has been published throughout the literature many times, and there are ways to do it with coronaviruses. Unfortunately, I think that game's going to continue to go unless people really learn that we're wasting a lot of effort testing people who don't get sick, and particularly the schools. And I've been really vocal about not doing this in schools because the kids don't get sick from this. And when you demand the PCR market focus on testing asymptomatic children, that comes at a direct expense of cancer patients not having access to testing. You may have noticed the COVID tests are like $100 to $300, right? PCR is normally a $30 test. It's only this way because we're mandating it and there's more demand than supply. So all of the reagents in the testing industry are getting vacuumed up into the COVID testing labs at the expense of cancer testing having access to reagents. So we're running around testing asymptomatic kids that don't get sick, and we're not able to test cancer patients because of this. So it's having a really massive impact on the supply chain in the testing industry. And it's just not spoken about enough that this monomaniacal focus on COVID is turning a blind eye to so many other diseases right now because of the weird incentives that are in place.
Yeah, I agree with you on that, and on the kids as well. I mean, from my understanding, and I want to fact check this, but I believe that since COVID began, it's been like twice as many kids have died of pneumonia as of
Oh,
COVID.
really? Yeah.
Yeah.
Or TB is
And
another one that comes up a lot, right?
what's that? Sorry.
TB is another one
Yeah,
people point to, you
yeah.
know?
And I think, and again, it's not like in the beginning was there was 0 deaths in kids, but I think it's still like less than 400 or 500 kids have died from COVID, so something like that. So,
It's less than flu. Yeah.
it does seem to be, or less than the flu. I mean, it does seem to be, and we don't make the flu shot mandatory for kids at school. So if more kids are dying from the flu than dying from COVID, I mean, and again, I'm not an advocate of making anything mandatory whatsoever. But if they're dying more of the flu than of COVID, then it would actually make more sense to give the kids a flu shot than a COVID shot in many ways. I mean,
Yeah.
some people will then say, okay, but the flu is not as infectious or is not as we could say infectious or as deadly as COVID. And therefore, you want to, decrease, asymptomatic spreading or that type of thing. I guess you could make that argument, but still, it seems like, overall, I don't agree with the fact that, kids should need or necessarily need to be vaccinated.
I'm very worried about that ethical slippery slope that we've gotten as a country in a position where we can say, under the guise of infectiousness, we can take the youth and sacrifice them for the elderly. I mean, what can't you do at that stage? Do you take their blood? Do you take their kidneys? Like what can't you basically redistribute from the youth upon the old? If the old are the ones that are at risk and you want to impose a mandated vaccination program on children who don't get sick, and arguably, I think the children have higher adverse response rates to the vaccines, at least the COVID vaccines, I should say. Where are you going with that? I mean, from a medical ethics standpoint, that doesn't fit any of the medical ethics I've written about or read about. That you can actually take one patient's health and sacrifice it for the benefit of another without their consent. I mean, that's
Yeah, I don't agree with that either. And Kevin, I know you only have a few minutes left, but I do want to ask you about one thing. So I haven't seen any real solid papers on humans with COVID with antibody mediated enhancement. But
right.
is that something that you feel is a concern with COVID? And for people who are listening, antibody mediated enhancement basically means that when you take the vaccine, that instead of and the way antibodies work for people who want to know is that the antibodies will mark the virus and the T cells come in and kill the virus or the cell. But basically what they're saying is that with antibody mediated enhancement, that's not going to happen, and it can actually, and by getting the antibodies from the vaccine, you can actually make your own response worse. So but again, and I know this is a real thing, it's happened with several vaccines in the past, but I haven't seen any human studies where they've shown this in COVID. Have you seen anything,
So
or are you worried
it's a
about this?
it's a really important topic right now, and so the antibodies can do two different things. I mean, they can neutralize the ability of the spike protein to get into another cell by binding to the spike protein in a way that inhibits it to get into the cell. But they can also, when they enhance the effect, sometimes the antibodies facilitate the virus getting into the cell. And if the spike protein continues to mutate, given the selective pressure it's under right now, a lot of us believe that when you deploy spike protein, really narrow vaccines that are non-sterilizing, like we have with the current vaccines, almost all the vaccines out there focus on just this 4 kb region of the spike protein, and the virus is 29 kb long, so it's really a narrow window of selection we're putting on the virus. So what's most likely going to happen is it's going to mutate the spike protein to get around the vaccine program that's out there, which there's some evidence that's happening now with the Lambda variant, they believe there's positive selective pressure there in the spike protein, and so this vaccine fact, I'd argue if you don't see selection against the spike protein, you don't have a vaccine program that's working. It should be putting some level of selection against that, and the sequences should change as a result of this. Ivermectin is a different story because you build immunity across the entire virus, but it has a whole different, we probably can't get into that today. My concern is I don't know if it's antibody-dependent enhancement yet from the data I've seen. There is a concern that there's neutropenia going on, that in the Pfizer trial, 46% of the patients exhibited neutropenia for a few days, and that lowers your white blood cell count, and that can make you more prone to infection. And then there's this myocarditis risk, which I don't think is necessarily ADE based. It could be something entirely different related to the spike protein and the concentrations that it's in when you're injected. It does appear as if the vaccinated cohorts have a higher rate of myocarditis than those who are naturally infected. I'll have to forward your folks some links for this, so I'm not accused of spreading this information here, but there are some papers out that are suggesting this. So I don't know why that's happening.
I have some concerns about that though too, because there is a real risk with it's not massive, but there is a real risk of myocarditis, especially with 2 shots. And now they're administering 3 shots with like basically no data whatsoever.
It's the same shot. It doesn't make any sense that they should shift it a little bit. It's like hitting the same antibiotic resistant bug with the same antibiotic.
I mean, it seems like it would only, make that side effect worse. I mean, if 2 shots, seem to be worse than 1 for elevating that risk, then, logically, you're probably thinking, well, 3 shots is going to elevate it even further. So, and I'm certainly not saying that if you're, severely immunocompromised, you don't get your 3rd shot, but like, if you're a fairly healthy person and you've already received 2 shots, I would hesitate about getting the 3rd one right now, just based upon that alone, because we really, really don't know the risk of myocarditis with a 3rd shot. I mean, there's no data on that like whatsoever.
There's no data, and we have a genetic form of AFib in our family, so I'm worried that we're an outlier, and that so these are measured, these rates of myocarditis are measured amongst the general population, and they don't genetically stratify them. Like, what do those odds look like if you have Brugada syndrome, or if you have some other form of genetic atrial fibrillation? I'm assuming they only get worse, but we don't know yet. So and it's like 6 million people, I think, in the country have AFib. So there's a large cohort of people that may have a pre-existing cardiac condition that isn't considered in those risk profiles. And if you have that additional prior, it may lean the odds even further away from taking a 3rd shot. And I don't know, I've been more impressed with the decentralized forms of medicine, I would say. When I look at ivermectin, there's 112 studies from all over the world with people that have different financial conflicts of interest, and hopefully all those things are neutralizing. And that to me is much more considerable evidence than the few studies we've had that have been heavily controlled by Pfizer. And even the Pfizer trial itself, they've kind of carved the data up in very clever ways to omit the people 14 days after the shots. All of the adverse events after the first jab are kind of thrown out. But if you go and dig into that data, you can see there's something going on there. Even in the Vax cohort, they're getting more cases of C19 after the first shot than the placebo arm. It was something like 400 to 200. So there's something going on after the first jab, whether it's neutropenia or some kind of immune suppressant that makes those patients more prone, has yet to be really teased apart, but it's in their data, and they're only really measuring the effectiveness after the 2nd shot. So there's a massive survivor bias going on when you do the analysis that way. I was reading someone's post today that was really, interesting perspective on this, that if you put a completely lethal vaccine through, you would have 100% effectiveness after the 2nd shot, right? Because the only people that survived, of course, were effective. So you have to look at both after the 1st and after the 2nd shot, and look at the data between all those. And that's kind of getting muddied right now. So there is a very bizarre monomaniacal focus on the vaccine program at the expense of any generic that could potentially play a role here. And that to me is the most frightening thing I've seen in the whole COVID pandemic, because the generic drugs are way cheaper, they're off patent, and you could use them prophylactically even. That's the study I'd love to see, is something like that versus the vaccination, to see who has more durable immunity. I suspect the immunity built up against 29 kb of the virus is going to be more durable than something built off of 4 kb of the spike protein.
Yeah, I agree with you on that, and then I'd love to chat with you further about it, but unfortunately we're out of time here.
Yeah.
So Kevin, you've been a wealth of knowledge here today, explaining a lot of things to us beautifully. So I think a lot of people are probably very interested in following you and following your work. So if you could just tell people your Twitter handle, where they can find you online, that'd be wonderful.
Sure. Kevin_McKernan on Twitter, and you can find me at medicinalgenomics.com.
Perfect. Okay, Kevin. Well, thanks for coming on, and we'll do round two soon.
Excellent. Thank you, Mike. Appreciate it.